Hi everyone, my name is Henry. I worked with Dr. Matthew Hare this summer here at Cornell, and I would sum up my talk as an oyster restoration effort along with a genetics reformation effort. Now before I run you through my research, I want to bring an analogy to you. Say you were given the task of figuring out where the particles of dust in this room originated from. So dust can originate from possibly the bed, the pillows, the rug, the couch, maybe from even outside. This was essentially my task this summer as I worked with oysters. Oysters are tiny little planktonic organisms early in their life, and they float around the water for around 2 to 3 weeks at the mercy of both, in my area of study, I'm looking at the Hudson River estuary. That's where fresh water flows into the saltwater Atlantic Ocean. And they're at the mercy of both this freshwater movement downstream as well as the saltwater tides going upstream. These oyster larvae also have a mind of their own. They're able to move up and down within the water column and essentially ride that fresh water or that salt water back in. They're also subject to lots of predation as well. An adult oyster has millions of oyster larvae that only survive up to a certain point, and lots of it become food for other organisms. And so how would we go about figuring where oyster larvae settles in this Hudson River estuary? Well, to figure out where they're going, we hung up these mesh shell bags full of oyster shells in the water since they like to settle on oyster shells. We did that on a 30-mile transect of river spanning from Hudson River Park all the way up to the Mario Cuomo Bridge. And this essentially allows us to figure out where they're going. Now, how do we figure out where they came from? There are two reproducing populations. There's a wild native population of oysters all the way up north, as indicated by that yellow star, as well as an aquaculture population that comes from an oyster farm that was placed into Hudson River Park down south. And we're using genetics to figure out whether they originated from this wild population up north or this restoration population down south. And that's essentially like scanning a barcode on an oyster. Now, when I mentioned the word genetics, I hope your mind didn't cloud over and had lots of jargon flashing in and out. And I also don't want you to be intimidated by the word genetics by thinking about things like genetic engineering or genetically modified organisms or even cloning. I want you to think more about the ecological perspective that genetics can bring to this work, especially with my work looking at population genetics between different populations in the Hudson River estuary. Now, my job is to balance the substance of genetics as well as make it simple enough for you to understand. I want to provide enough content to allow you to understand the impact that genetics had on my work, as well as make it simple enough for you to not get bored by my explanation. Now, I could present you with this principal component analysis chart in which 274 different genes are graphed in 274 different dimensions, but that would probably bore you very quickly. I could also present you with this rainbow double helix and tell you that I use genetics in my work, but that probably wouldn't do you very well either. And so I'm going to use a more significant diagram showing you how oyster larvae, although it's not this linear, they essentially migrate from their source population to our shell bags. We take those oyster larvae, extract DNA from them, and from this DNA we can genotype the oysters to figure out whether they came from this northern wild population or this southern aquaculture population. And so why do we want to know where oyster larvae are coming from and where they're going? This helps us inform genetics by placing oyster restoration habitat where oyster larvae are going. And we can also place oyster populations that are reproducing in habitat that isn't seeded by many oyster larvae. Now, we want to restore oysters to the Hudson River estuary itself, not only because they're native, but because they also provide very valuable ecosystem services. You can see here, oysters filter up to 50 gallons of water a day, which helps to clarify the water and take toxins out of the water column. They also provide great habitat for organisms, thereby increasing biodiversity, and the reefs help to absorb wave action, which can mitigate flooding during storm surges. Now, when I talk about oysters to the general public, there's a mix of oysters are nasty, they're slimy, they're undesirable, snot on a rock essentially. And others, when they think about more charismatic organisms that need our help, they think about animals like seals and sea lions, in which I worked with last year. And I can really see that difference in the public support that people have for the organisms that I've worked with, both current and in the past. And so the way people interact with marine organisms is essentially impacted very heavily by pictures, but also, I argue, with stuffed animals. And so we see a regular oyster here, pictured above, relatively charismatic, as I can argue. We also see a oyster stuffed animal, not too sure why it has a face, but it's also blushing, and it has arms and legs, which I really don't know where those came from. And finally, we have our last stuffed animal. The pearl has a face. I don't know why the pearl has a face, but we really do anthropomorphize things that we don't perceive as traditionally charismatic to allow the public to relate to them. But I argue that a better way to advocate for oysters is by introducing people to oysters themselves. I worked with Hudson River Park this summer, as well as their River Project Wet Lab down at Pier 40. That's in the southern region of Manhattan. And every summer they open their lab up to visitors and display live organisms that are native to the Hudson River. And many people are often surprised by this because they perceive the Hudson River as something that's dirty and lifeless. So this really surprises them. And we rope them into our work as well by training volunteers to help us comb through our shell bags in order to find oyster larvae on these oyster shells. And so I leave you now with 2 oyster larvae that I found this past weekend. They can range from the size of an ant to the size of 2 or 3 quarters in area. And whenever you come across a study that talks about genetics or that informs you about the potential possibility of using genetics for a certain study, I want you to stay open-minded. Genetics can have huge implications for all aspects of work. It can answer questions that we can't even fathom yet. And so the potential that genetics has for informing, especially restoration work here, in my case, is limitless. And one more thing, if you're ever in the market for an oyster stuffed animal, skip the face, skip the arms and legs, and go for something more anatomically correct. All right, thank you.